So thank you very much for joining today's Open Box Science seminar that will be about cancer, in particular about the role of macrophages and in different kinds of cancer. So it's my pleasure to introduce you to the first speaker, Dr. Nicoletta Caronni, author of the paper IL1beta macrophages fuel pathogenic inflammation in pancreatic cancer. She's currently a senior postdoc at San Raffaele Teleton Institute for Gene Therapy in Milan, Italy, and she will be presenting us the work they do there. So with that, I would like to remind everyone that this is usually a talk of around 20 minutes or so, which is followed by questions and answers. You can type your questions during all the time in the chat, and you can also ask them live later. So with that, thank you very much, Nicoletta, for joining us today, and I'm looking forward to your talk.
Thank you, Anna. Thank you for the introduction. So can you confirm that you see my screen?
Yes, perfectly.
Okay, great. So thank you for the kind invitation, and I'm really happy to be here today to share with you our latest results on the IL1beta positive macrophages. How we found, I will share with you how we found this subset of macrophages that is able to fuel the pathogenic inflammation in pancreatic cancer. So I will start with a, if I can, because okay, with a brief introduction. You do see here also this parted, right?
Yes, we see.
Okay, I will start. I let you know and I've decided why we decided to look at the macrophages in pancreatic cancer. So first of all, our lab is mainly focused on studying the biology of macrophages, and in particular to understand how macrophages respond and sense inflammatory stimuli. So as probably all of you know, macrophages are tissue resident cells of the immune system. And that it exerted many different functions according to the tissue in which they reside. But the common goal of these complex functions is actually to support the physiological functionality of the organ in which they are present, and to keep and maintain the homeostasis of this organ. Unfortunately, under pathological conditions and mainly during cancer progression, these functions are actually co-opted by the tumor. And at the end, the macrophages end up to support the tumor itself instead of supporting the healthy tissue and the functionality of the healthy tissue. In the last years, it has been, thanks to the single-cell RNA sequencing, it has become very clear that tumor-associated macrophages are not an homogeneous population, and in particular, many subsets of transcriptionally defined macrophages have been described in the literature, and I reported here some of them, like macrophages expressing FOLR2 and LIPE1 that resemble the tissue-resident macrophages, but also other subsets of macrophages have been described, like macrophages expressing interferon-regulated genes, like CXCL9, proliferating macrophages, and also macrophages enriched in lipid metabolism genes. However, the functions of these subsets still need to be addressed and well understood. And this is, we think, it's a critical factor because we think that understanding the functionality, and in particular which subset of macrophages are anti-tumor or pro-tumorigenic, is very important to design novel immunotherapies in order to selectively address and target a specific population of macrophages. So we decided to look at the heterogeneity of the macrophages in the context of pancreatic ductal adenocarcinoma, that you probably know is a deadly disease with poor, very extremely poor prognosis and only few therapeutic options. And moreover, the pancreatic ductal adenocarcinoma is actually characterized by the abundance of the macrophages and also by an inflammatory tumor microenvironment. And in the last years, many papers came out in the literature describing the importance of the inflammation in PDAC, not only in the established PDAC, but in particular in the early phases of tumorigenesis, where inflammatory signals have been described to cooperate with oncogenic mutations in order to accelerate and facilitate the neoplastic transformation of the epithelial cells. So in this context, we wanted to start to decipher the heterogeneity of the tumor-associated macrophages in order to describe the transcriptional programs and the factors that are able to drive the pathogenic activities of these macrophages, with the final goal to identify possible novel targets for immunotherapies that are currently ineffective in PDAC patients. So we started by performing single-cell RNA sequencing of human PDAC samples from naive and chemotherapy-treated patients. And we retrieved 6 transcriptionally defined clusters of macrophages. Some of them have been described in literature, like the FOLR2, the SPP1 positive macrophages, and also the proliferating macrophages. But we decided to focus our attention on this cluster here, cluster 1, that we named IL1beta positive macrophages. These cells are enriched in genes related to inflammatory response, leukocyte migration, cellular response to LPS, but at the same time, we found out that they are depleted of genes related to type 1 interferon response and antigen presentation via MHC class 1, so indicating that this IL1beta positive TAM could be probably associated more to a pathogenic type of inflammation rather than a cytotoxic type, because we know that type 1 interferon and the presentation via MHC class 1 are very important to elicit a proper anti-tumor immune response. So to test this hypothesis, we decided to look at the TCGA cohort of PDAC patients, and we stratified these patients according to the expression of the IL1beta positive TAM signature. This signature is composed by 6 genes that are specifically expressed by the IL1beta positive TAM and not by the other TAM subset, but also they are not expressed by the other cell types that are present in PDAC samples. And as you can see here, we observed that the patients with the higher expression of IL1beta TAM signature show a worse prognosis as compared to the patients expressing low levels of the IL1beta positive TAM signature. So in the end, we observed that the IL1beta positive TAM cluster that we identified are probably associated to a pathogenic inflammation, and they are also associated with the poor prognosis in PDAC patients. So to understand better the biology of the IL1beta positive TAM, and in particular, we wanted to understand whether these IL1beta positive TAM could originate from the circulating monocytes or from the tissue-resident macrophages. We decided to use an orthotopic model of PDAC. So we use a cell line, a KPC cell line, that is derived from the genetically engineered mouse model, which holds a mutation in KRAS and p53. And we injected these cells directly into the tail of the pancreas. And to understand the origin of these macrophages, we generated a dataset of transcriptomic profile from monocytes and macrophages coming from control pancreas and orthotopic PDAC at different time points. So the 10, 20, and 30 post-tumor inoculation, and also the monocytes from the blood of these animals. From this data, after the integration of all these transcripts, we performed cell rank and RNA velocity analysis, and cell rank identified the IL1beta positive TAM macrophages as the terminal state of a trajectory that starts from the circulating monocytes, it goes through the tumor-infiltrated monocytes, and it ends up in at the IL1beta positive TAM, indicating that the IL1beta positive TAM are likely from the monocytic origin, and they are not derived from the tissue-resident macrophages. Moreover, when we look at the expression of the marker genes of the IL1beta positive TAM or the positive TAM in both the monocytes that are represented here in light blue and the macrophages, the IL1beta positive TAM, that are here in red, we observed that monocytes, either in the circulation or in healthy pancreas, they express very low levels of the IL1beta positive TAM signature, but the signature starts to be upregulated as soon as the monocytes get into the tumor, so 10 days post-tumor inoculation, and is actually highly expressed, further expressed at day 20 and 30 post-tumor inoculation. So this data indicated that there are some local factors that are present at the tumor site in the tumor microenvironment that are able to recruit the monocytes at the tumor site, but also they're able to differentiate those monocytes into IL1beta positive tumor. Obviously, we wanted to understand which are these local factors. And to make a long story short, we identified prostaglandin E2, which is a lipid metabolite, and the TNF-alpha as the main driver of the IL1beta positive tumor state in PDAC. These two factors are abundantly enriched in both mouse and human PDAC as compared to the control tissue. And moreover, when we stimulated bone marrow-derived macrophages and monocytes isolated from the bone marrow in vitro with the TNF alone or prostaglandin E2 alone, you can see here in gray and in blue, we didn't see a strong upregulation of the IL1beta expression. However, when we co-stimulated both cells, so macrophages and monocytes, with prostaglandin E2 together with TNF, that is actually a condition more resembling what is happening in the tumor microenvironment, where multiple factors are present in the same time in the tumor tissue. We did see a dramatic induction of IL1beta. And this is true not only for IL1beta itself, but also for other genes that we identified by bulk RNA sequencing, and we called the PGE2 and TNF-alpha synergized genes. And we observed that these genes are actually selectively enriched only in the IL1beta positive tumor and not in the other tumor subset, indicating that prostaglandin E2 and TNF-alpha are the drivers of the IL1beta tumor state. Obviously, these analyses have been performed in vitro, so we decided to look also in vivo what happened and whether prostaglandin E2 in particular is fundamental to driving the IL1beta positive tumor state. So in our context, prostaglandin E2 is mainly produced by the cancer cells themselves. So we decided to generate COX-2 knockout PDAC cells. COX-2 is the main enzyme that metabolize the arachidonic acid into prostaglandin E2. And we injected these cells together with the control cells, so PGE2 proficient PDAC cells, into monocompetent mice. And as you can see here in light blue, COX-2 knockout show a dramatic reduction in the tumor growth. So they can establish, but then the majority of the tumors are actually rejected. So in order to look at the tumor-associated macrophages in this context, we decided to perform single-cell RNA sequencing at very early phases of tumor progression, so 7 days post-tumor inoculation, when there is comparable tumor masses between the two conditions. And obviously, we focused on the transcription profile of the macrophages, and we observed several differentially expressed genes between the presence and the absence of the prostaglandin E2 in the tumor. And in particular, we observed that COX-2 knockout tumor-associated macrophages are depleted of the IL1beta positive tumor marker genes, whereas they are enriched in type 1 interferon response genes. So indicating that the absence of the prostaglandin E2 is associated and favor the cytotoxic profile of the tumor-associated macrophages. And instead, the prostaglandin E2 produced by the cancer cells is essential for driving the tumor progression, the tumor growth, and also the development of the IL1beta positive tumor. We then wanted to understand whether the IL1beta itself had a pathogenic role, a pathogenic function in PDAC growth. And so we decided to perform a very simple experiment by neutralizing IL1beta along with the tumor progression. And as you can see here in blue, we observed a dramatic reduction in the tumor growth when we depleted IL1beta. And we also identified the cancer cells as the major cellular target of the pathogenic activity of the IL1beta in PDAC context. Indeed, when we generated the IL1R1 knockout tumors, IL1R1 is the signaling receptor for IL-beta, we observed a dramatic reduction in the tumor growth. And this growth is actually restored when we representated the expression of the IL1R1 in the knockout cells. So this data indicate actually that the IL1beta signaling in the cancer cells is essential to promote the tumor growth. What happened after the stimulation with IL1beta in the cancer cells? We stimulated the cancer cells in vitro, and we performed both bulk RNA sequencing and multiplex ELISA, and we observed the induction of multiple genes of the inflammatory response after the stimulation with IL1beta of the cancer cells. And in particular, we observed an induction and even higher production of prostaglandin 2 and TNF that, as I already told you, are the main driver of the IL1beta positive state. So at the end, we end up defining a cellular loop, a self-sustaining loop, where the IL1beta that is produced by the IL1beta positive macrophages is able to induce an inflammatory reprogramming of the cancer cells that upregulate the production of PGE2 and TNF alpha that further sustain the IL1beta state and this inflammatory loop. We then define a more precise tumor intrinsic IL1beta response signature, aka T1RS, by generating transcriptional profile of multiple PDAC cell lines, cultured both in 2D and 3D. And we decided to look at the expression of these genes in a TCGA cohort of PDAC patients that we have already certified for the expression of the IL1beta signature. And we observed that the patients that showed higher expression of the IL1beta signature, meaning that have a more abundant content of the IL1beta positive TAM, show also higher expression of T1RS signature. And we also observed that mainly the genes that are part of the T1RS are associated with poor prognosis in PDAC patients. We also took advantage of publicly available data sets of healthy epithelial cells and tumor cells from both human and mouse PDAC. And we observed that the T1RS genes are mainly enriched in PDAC cells, so in the tumor cells, as compared to normal adjacent to epithelial cells or epithelial cells from a healthy mouse. So T1RS is specifically expressed in the tumor cells and not in the healthy epithelial cells. And we confirmed it also when we looked at this data set in which there is a transcriptomic profile of epithelial and tumor cells in different stages of PDAC progression. And as you can see here, T1RS is not expressed in the healthy pancreas, but it starts to be expressed as soon as there is the oncogenic activation of KRAS. And then it's further upregulated in early and late PANIN that are precursor lesions of PDAC. The expression of the signature is then maintained in the established PDAC and also at the metastatic site. But these data are telling us that T1RS is not important only in the established PDAC, but also is associated with the oncogene activation in very early phases of tumorigenesis. So finally, we came back to the single-cell RNA sequencing of the human patients that I showed you in the first slide. And this time we focus our attention on the tumor cells. So we generated a reclustering of the tumor cells, and we look whether these cells were able to did express the T1RS. And as you can see here, we found that only some clusters in this clustering are expressing the T1RS. And we also identified that cells that are expressing the T1RS are at the endpoint of a pseudotime trajectory. And generating profound gene set enrichment analysis of the genes that correlate with this pseudotime, we observed an enrichment in genes associated with pathways that we know are related to IL1beta signaling, like TNF alpha, signaling via NF-κB, hypoxia, inflammatory response, ROS production. But we also observed an enrichment in genes related to EMT, KRAS signaling, TGF-beta signaling, p53, that are all pathways that are related to malignancy and tumor aggressiveness. So These data indicate that the expression of the T1RS in the cancer cells is correlating with the acquisition of the pathogenic signatures. Finally, for the last part, we were really intrigued by the fact that only a subset of macrophages is able to produce IL1beta, and we found out that only a subset, a population of PDAC cells was able to sense the IL1beta. So we started to wonder whether this inflammatory loop, self-sustaining loop, was mainly supported by a proximity, a local proximity between these two populations. And to answer this question, we decided to use a spatial gene expression approach on the PDAC samples, the human PDAC samples. And we decided to use the molecular cartography, which is based on in situ RNA hybridization, and it allowed the subcellular resolution of a transcript of 100 genes. We defined this panel based on our single cell RNA sequencing in order to select the genes that allowed us identifying the major cell populations that are present in the PDAC samples, and obviously also the subpopulation that we are most interested in, that are the T1RS positive PDAC cells, here the cluster 13, that express, as you can see here, markers of tumor cells, but also 6L1 that we use as a proxy of T1RS, and the IL1beta positive TAMs that express the marker of macrophages and also IL1beta itself and NLRP3. So with this data, we perform spatial enrichment analysis. So the analysis of the neighborhood of these IL1beta positive TAM and T1RS. Here I reported the output of the data, but here I also reported for you a representative image of a patient in which we found the IL1beta positive TAM and the T1RS positive PDAC cells that are represented in red and in light blue respectively, and these two are the two magnification of this ROI. So how we did this analysis, we starting from the IL1beta positive TAM, we took each IL1beta positive TAM and we look in the surrounding areas with different diameters up to 40 microns, and we analyze the cells that are present in these areas. And we found out that the population that is actually mostly enriched in the neighborhood of the IL1beta positive TAM are the IL1beta positive TAM themselves, indicating that these cells are localized as clusters in the samples, and you can also see a nice representation here in the magnifications. But the second population that is mostly enriched in the neighborhood of the IL1beta positive TAM are the T1RS PDAC cells. Conversely, analyzing the T1RS positive PDAC, we observed that also in this case, the cell population that is mostly enriched in the neighborhood of the T1RS positive PDAC cells are the T1RS positive PDAC cells themselves. But the second population is corresponding to the cluster 16 that are the IL1beta positive TAMs. And you can see here very nicely how these two clusters co-localize together. So at the end, we identified specific local areas of the tumors in which tumor cells and IL1beta positive TAM can influence each other, fueling the pathogenic inflammation and the progression of PDAC. So in the end, I have many people to thank. First of all, Renato, that supported me and mentored me during these years, but also many people, these people here that contributed with me to this work, in particular Francesco, a very talented PhD student that shared with me the last last 3 years in generating all this data, and Federica and Giulia, that are the two amazing computational biologists that had the duty to analyze all the transcriptomic data that we generated, and also the other person in the lab that contributed to this work, Marco, Simona, Luca, and all the collaborators. And obviously, thank you for the attention.